Before we can talk about why this is important, I've got this simple explainer. Inside a human cell, there's a power source called the mitochondria, which contains DNA. That DNA is passed to offspring, including faulty DNA that can cause devastating genetic diseases. Now, local scientists are discovering ways to splice up that DNA so a parent only passes along the healthy genes. Here with the details are my guests, mitochondrial DNA researcher Alex Ocampo with the Salk Institute and Evan Snyder, an FDA advisory board member for a similar DNA technique with the Sanford Burnham Medical Research Institute. And Alex, what kind of diseases or conditions are caused when bad mitochondrial DNA is passed to an offspring?
So mitochondria is involved in essential functions in the cell. One of them is energy production. Maybe the most important one is energy production. When you have mutations in mitochondrial DNA, these diseases will affect mainly organs such as the brain, the heart, the muscle. So these will be very devastating diseases affecting kids that will suffer from muscle weakness. They have a brain impairment, neurological disorder, liver disorders, many other disorders, and they actually reduce quite some the life expectancy. So these kids will only live into the early teens, and many of the cases it will be fatal diseases.
And it's very disabling, this particular types of genetic diseases. Now, Salk is doing research. You're doing research on mice to eliminate the faulty DNA, but still get the mitochondrial good DNA into the offspring. So in the simplest terms, can you tell us how you're doing that? How do you take out the bad DNA and just leave the good DNA?
Okay, so what we do is we take advantage of the situation in the oocyte or in the embryo in which you have wild-type normal mitochondrial DNA and mutated one. What we do is we introduce some molecular scissors that will go and will recognize which ones are mutated, which one is the bad mitochondrial DNA, and will make a cut in that molecule. That will lead to the degradation of the molecule, and that will mean that the kid that will come from that embryo will be a healthy kid. So
far, like you said, you've been doing it in mice. You have these scissors, these molecular scissors that cut out the bad DNA. How's the research been going? Have you been successful?
Yeah, actually, we were very happy with our results. So we are using a mouse model in which you have two types of mitochondrial DNA, and what we aim is to prevent the transmission of one of them to the next generation. So we use these molecular scissors at the level of oocytes or embryo, and actually we saw that we were able to prevent the transmission to next generation, and not only to the first one, but to the generation, the third and fourth one.
So the little mice come out okay afterwards. Yes. Without this. Now, Evan, are there ethical concerns with the type of research going on here at Salk, messing with the DNA, the mitochondrial DNA in mice?
Well, I think the most pressing ethical concern, other than dealing with manipulation of the genome, is whether or not you are going to introduce unintended changes, create other diseases, change traits that were unanticipated. That's one area. And then the second is, could you influence subsequent generations and create something that you did not anticipate, but now gets carried in humankind, so to speak, if this were in humans.
So bringing it back to the whole human race. Now, I know in China, scientists tried something similar, not exactly with nuclear DNA in human embryos, but the embryos weren't viable. Unlike yours, these embryos couldn't reproduce and didn't go on to be viable. But that whole experiment in China caused quite a backlash and quite an ethical uproar. What is the issue a little bit more as far as when you're dealing with human embryos?
Well, first of all, in dealing with human embryos, one is dealing with the roots of human life. The experiment in China probably would never have been sanctioned in this country. And I don't think scientists here are endorsing the fact that that experiment was done. However, one always tries to find something instructive, even in an experiment like that. And the lesson we learned was that, in fact, our fears were confirmed. That, in fact, there were off-target effects. There were mutations that were unintended. And that had this been done, it would have created offspring and maybe even a lineage that would have been horrible and terrible. So we've learned that, in fact, we need more science before we could ever move on.
Sure, and the science is like what you're doing, Alex. So are there next steps for the mice embryo or other embryos how will you continue in this research?
Okay, so obviously the next step will be to move to humans. So we plan to use oocytes from mitochondrial disease carriers or also discarded embryos from IVF treatments. The idea is mainly to prove safety, so we want to make sure that this technique is safe. So we will go ahead and do some research with human samples just to verify that the technique can be used in a safe way. And that, I guess, will open the door to the next step that we'll try to see if the technique can be applied in the clinic. So you have to demonstrate that the technique can be good use to eliminate the mutated DNA in the human embryo. So that will be, I guess, next step. But first we go with safety and make sure that the technique doesn't have off-target effects like in the Chinese.
So we have a few years off here before we see
Yes.
any results in the human population
Yes.
on this. All right, Alex Ocampo and Evan Snyder, thank you so
Thank you.
much.
Thank you.